disease. Let's start the vaccine. So first of all, we need to know before we start the vaccine, the technical name from immunology. That what vaccines we use to say. Artificial active acquired immunity. Artificial active acquired immunity. Technically, we call vaccines as artificial active acquired immunity. So what is the mechanism? We introduce the microbes. Generally, when they come naturally into our body, we frame the sentence. Once we get the disease, we don't get it for second time. We develop memory cell. So now what do you want here? We should not get the disease, but we need to develop memory cell. How? How is it possible? By taking the dead microbes, by taking the weak microbes into our body, that we develop, they're just playing practice match. Okay, so by taking the dead microbe, by taking the toxic product of the microbe, or by taking the killed microbe, we develop memory cell without getting the disease even for first time. What you call technically artificial active acquired immunity, which is termed as vaccine. Then we say it as vaccine. Okay, so this is about the vaccine. We have some types of vaccine. Let us see that vaccine. So is given this vaccine, Edward Jenner. The term vaccine was given by Edward Jenner, coined by Edward Jenner. Okay, he immunized a boy against the smallpox by inoculating him with a relatively less dangerous cowpox virus. That smallpox is a famous disorder older days. That the like corona it new nowadays, smallpox is also killed so many people, but those who survived even they'll get the post smallpox disorders like the depressions and the skin, this kind of bigger problem it is actually in the days. But there is behind a lot of bigger story actually. The people who working in the dairy farms are not getting this particular smallpox. Why? So when they started identifying, Edward Jenner started discovering. So like we have smallpox, there is a cowpox virus which will come to the viruses, to the cows. That same virus, that our cousin brother, smallpox, like cousin of smallpox, there is a cowpox which is giving a weak virus in compare with the human being. But that is able to cause smallpox in cows. Okay, so the technology, actually we know the complete technology. That's why let's talk about in the same technical way that when a particular in dairy farm people when they start closely moving with while taking milk, withdrawing the milk from the cow, they move closely with the cow, isn't it? So that means they are infected with cowpox virus. As cowpox virus is so weak, cowpox virus is so weak, that's why they are not able to get the smallpox, but they developed memory cells. Okay, they developed memory cell. We are not fighting with the hero, we are fighting with the dupe of the hero, but we are able to develop memory cells. Now we can fight with the hero also now. So that's the thing happened. Okay, so that was the observation made over the discovery of vaccine. That observation. Okay. So the technique of attenuating means live or weakening of a microbe was developed by Louis Pasteur. Okay, we have so many stories here. They introduced in boy, or some says they injected on himself that smallpox. First he injected cowpox, and after smallpox virus injected on himself. These kind of stories, but we got success actually. We got success. What's the success? That a technology, a new technology came because of that observation that smallpox is coming all over the world, but those who are working closely with cows, that in those people they are not getting. So that information made us to get the doorsteps of the vaccine we entered. A vaccine is a biological preparation that improves immunity to a particular disease. A vaccine typically contains the disease-causing microorganism. A vaccine typically contains the disease-causing microorganism. That is very important line. In the any vaccine, in corona vaccine, what we will find? We will find coronavirus. A vaccine typically contains the disease-causing microorganism, but often made from weakened or killed form. Okay, when we give the take in the killed form, that is little bit problematic. In what way? The killed form don't generate more number of memory cells in our body. Okay, in that way, if you take active form, it may die even. That's the problem actually. The toxins are one of the surface proteins of the microorganism. That means sometimes we don't take the whole microbe. We'll take the toxic product released from the particular microbe. Toxin are one of the surface proteins of the microorganism, also used as preparing vaccines. Okay. So whatever it may be, giving information before that original hero will come. We can't say hero, we say villain, isn't it? Vaccines are generally used as a prophylactic measures. That means, hoping so in future when I get the disease, I will develop memory cells that make us to be protected. Moreover, they may not guarantee the total protection. It doesn't mean like, so if you take the vaccine, you may not get the disease. That means how far the memory cells you are developed, that depends, isn't it? It may be due to inadequate immune response. How your immune system is reacted, how you develop the memory cells, that is also very important here. Okay, it is person to person, it may be varying. Even if the host develops antibodies, the human immune system may not effectively defend the body from the pathogen immediately. That's the story we're talking about. Therefore, certain adjuvants, sometimes we don't respond. That I said over. Those who don't attack more, we don't respond against them. That's why we don't develop memory cell. For example, a virus you are taken into the body, dead virus, fully dead virus, you are taken. That enters in the body. We respond, but adequate response may not be possible because the killed one. That remember that story I said over. A person entered in the house, not saying anything, started destruction. We defend him actively. A person entered, even you don't know him, a person entered and just sitting, not doing anything. So we respond against them, but adequate response we don't show. That's the thing you have to remember. So therefore, certain adjuvants along with the substances of the dead microbe, we have to use some adjuvants. What are adjuvants? Those who enhance the immune response stimulated by the antigens. We call them adjuvants. Generally, aluminum adjuvants will be used over. Okay. So that means along with the dead killed microbe, we are giving some adjuvants also. Okay. So use of vaccines in Afro-Asian countries has reduced the mortality rate. For example, smallpox. Smallpox could be eradicated completely. Nowadays, you people not listening about the smallpox, isn't it? Now it reached to general knowledge books. The disease which is eradicated completely from India, smallpox. Okay, that intensive vaccination program in an organized manner, that by governmental agencies, as it is being done in the case of polio also nowadays. Hope so in future time, much reduced over polio. So because of that, we can announce in future time that polio is completely eradicated. Very, very less cases are present in India for polio. Because of the vaccine, all it happened. Hope so. The same way, the coronavirus, COVID vaccine also will come in future. So there are diseases such as AIDS and malaria are yet to be developed. I should add one more, isn't it? Corona, coronavirus. Why we not developed AIDS and malaria? We have answer here. You know that AIDS and malaria. Why we are not able to develop till the time that vaccine against malaria? I want answer from you. Vaccine against malaria and AIDS. Why we are not developed? Same answer will be applicable for corona also nowadays. I think you people may know that. Just send the message to me. Why we are not able to develop? I want detailed description. Okay, AIDS and malaria. Why we are not developed? The same answer will come for corona also. Why we are going to late? Okay, so let's continue with the vaccines. So nowadays, the program is going on for the preparation of vaccine of corona. Why we are late? That the same answer, whatever the answer we have for AIDS and malaria. So try to find out the answer. Now let's go with attenuated whole agent vaccine. Various types of vaccines are there. Very few we are learning now. Attenuated whole agent vaccine, inactivated whole agent vaccine, toxoid vaccine, attenuated whole agent vaccine, and the toxide vaccine. We have more recombinant vaccines are there, DNA vaccines are there, likewise various types of vaccines are there. Attenuated whole agent vaccine. So that means what material we are using, based on that we classify. In case of attenuated whole agent vaccine, they contain the disabled, less virulent, we can say, live microorganisms only will take, but not like original. So mostly they are antiviral, these kind of vaccines. Examples, vaccine against live only, but less virulent, less harmful. Okay, so we develop in laboratory. Vaccines against yellow fever, measles, rubella, mumps, and the bacterial diseases such as typhoid, we are using attenuated whole agent vaccine. Inactivated whole agent vaccine is there in the name. Inactivated, the name indicates dead. Whole agent, the word indicates the total microbe in the dead form. Attenuated, whole microbe in the live form, weakened form. Then inactivated whole agent vaccine. Total microbe we are taking inside, but in the dead form. Virulent before killing. Killed microbes are taken out. Okay, so example, we have so many like vaccine against influenza, cholera, bubonic plague, polio, hepatitis A, rabies. Then in case of polio, that Salk polio vaccine is used locally. Then toxide vaccine, we don't take the microbe totally. The product of the type, that particular microbe or surface protein of the microbe. They contain toxides, which are inactivated exotoxins of the certain microbes, inactivated toxic material. So exotoxin means where the microbe release the product, that kind of toxin, directly not that, will take that inside to develop for what? All the three for develop for what? To develop memory cell. This is very important. To develop memory cell. Whatever the vaccine it is. Typhoid, tetanus and diphtheria are the vaccines, the toxide vaccines we used to say. Again, is the tetanus and diphtheria. And these vaccines are used in the prevention of diseases as they induce artificially acquired active immunity. So technically what I said is as artificial active acquired immunity. Finally, a new development in the case of vaccines are about polio vaccine. Earlier I said over it, we are using inactivated whole agent like Salk polio vaccine, which is given by Jonas Salk. That he introduced the inactivated polio vaccine, which became more popular as Salk's vaccine. But Sabin is given, Albert Sabin is given oral drops. Nowadays we are taking oral drops, isn't it? What is this oral drops? Attenuated whole agent vaccine, that is. That means live but less virulent, produced by Albert Sabin. So current time, we are taking drops, polio drops. That is Sabin's vaccine. We see Sabin's oral polio vaccine. We used to say became much more popular as it effect lasts longer and easy to administer. Okay, so that's it. Attenuated whole agent, inactivated whole agent, and toxide vaccine, and change in the vaccine, polio vaccine. Earlier we are taking Salk's inactivated whole agent vaccine, but nowadays we are taking Sabin's oral polio vaccine, that is which is attenuated whole agent vaccine. So that's it about the vaccines.